The process of DNA replication is crucial to the overall reproduction of a cell. And so anytime a cell is going to reproduce new copies, it is going to have to first copy the DNA molecules and ideally make exact copies of those DNA molecules so that it can really place those in the new cells that are being produced. So what we see here is a look at the overall process of DNA replication. One of the benefits of having a double-stranded DNA molecule was that if you look at that molecule, you've got two strands that are essentially opposites of each other, or what we call complementary. So they have the opposite base pair in them. So we'll start with a double-stranded DNA molecule here, shown in purple. And through the process of DNA replication, those two molecules, those two strands are going to get essentially pulled apart from each other. So they'll be peeled apart, and then each one of those halves that we have is going to serve as a template to build a new copy of DNA right there on top. So if you look at this first replication that we have, you have one purple strand, which is your old DNA molecule, and that's going to be connected to one green strand, which will represent your new DNA molecule. And we'll have two copies like that, and we expect that both of those double-stranded DNA molecules are really exact copies of each other, so identical in every way. And then if we were to go another round of DNA replication, you can see that we would take that molecule, which is half purple and half green, we peel that apart, and we're going to build a green one, a new one, on top of both of those. Well, what we're seeing here is what we refer to as the semi-conservative model of DNA replication. So that is the accepted model, and that model is specifically stating that when we perform this process, we end up with a double helix, a double-stranded DNA molecule that has one old strand of DNA connected to one new strand of DNA. So our molecules are basically half and half after replication. As far as how the whole process is going to take place, that complementary strand of DNA, the opposite or opposing strand of DNA, is going to be synthesized. It's going to be made using the base pairing rules. So just to remind you, the base pairing rules are that adenine is always going to hydrogen bond with thymine, so we're going to find them across from each other. And just to sum it up, A and T, they form two hydrogen bonds with each other. And then down here in the bottom picture, you can see that guanine and cytosine, they are going to hydrogen bond with each other, and they're always going to form three. So across from every A in this strand, we're going to find a T in this strand. And across from every G, we're going to find a C. So that's the complementary part of it. So if we look at what we end up with, here's the double-stranded DNA molecule, and you can see that we always have those base pairing combinations all the way down the end side. So what that means is that if we know the sequence of one of these DNA molecules and we know the base pairing rules, and there are molecules inside the cell that know those rules, then we can take this template and we can just build the other one right on top of it. So that's the nice thing about having this double-stranded DNA molecule with the base pairing rules. All we need to do is look at one of the strands and we should be able to come up with what the second half of it ought to be. So if we talk about how the process is going to take place, the energy for this process, this is an anabolic process because we are making DNA, the energy for this synthesis is contained within the nucleotides that we're actually going to be using and joining onto this overall growing polymer. So here's a look at one of the nucleotides. This is a nucleotide triphosphate. So this happens to be deoxy-GTP. And this is very similar to an ATP molecule. So this is really referring to the fact that you've got these bonds here, which are very unstable. And because they're very unstable, they are high energy bonds. And high energy bonds, if you break them, energy comes out. And that energy that's released is used for this overall building process. So if we look at it down here, basically we're going to take an existing molecule or strand of DNA, and then we're going to add to that a nucleotide triphosphate. We'll cut off two of those phosphates. So if we just draw it, basically we're cutting this off. That's going away, and then we will have and then we will have the newest nucleotide added onto our growing chain. That's the process of extending this molecule and making it bigger. So So if we talk about how this is going to happen inside the cell, well, the first thing that has to take place if we're talking about DNA synthesis is that there has to be an overall unwinding or peeling apart of the DNA strands. And this unwinding or peeling apart, this separation, is going to happen at a location which we refer to as the origin. So the origin is our start site. So I'm just going to put that right here, that this is the start site, starting location. And the peeling apart of the two strands is going to occur with an enzyme called DNA helicase. So what DNA helicase is going to specifically do is it is breaking the hydrogen bonds that hold the strands together. So we had lots of hydrogen bonds all the way down the middle of this double helix, and this helicase enzyme is kind of like a bulldozer is the way that I think of it. It's basically ripping through there and breaking, popping all of those hydrogen bonds that are holding the strands together. Once we do this, we are going to have what we call a replication fork. And the replication fork is just the specific region where we have peeled apart and separated these strands from each other. Another thing that we have here is stabilizing proteins. The stabilizing proteins, their job is to keep the separated strands separated. So these bases that we have in the two strands, they would love to just snap right back together again once they've been peeled apart because they are attracted to each other. So the stabilizing proteins, if you look at them right here, they are sitting right on top of these single-stranded DNA molecules and preventing them from snapping and closing back up. When the process of DNA replication actually gets underway, so once we've got the origin all opened up, the enzyme that's going to be synthesizing and actually making the new DNA is called DNA polymerase. And that's the enzyme we see right here. Well, DNA polymerase is going to read the DNA template. And as it reads the DNA template, it is going to make the new DNA. So it's the main enzyme. If there's one enzyme that's doing most of the work, it would be DNA polymerase. So if you look in this picture here, DNA polymerase is kind of the pink structure, the pink enzyme that you see kind of wrapped around the template DNA. It knows the base pairing rules. So if it sees a T, it knows to put an A across from that. And that's what you see it doing here. Or if it sees a C in the old strand, it knows to put a G across from it. So as it goes through, it's going to basically keep adding to the end of that molecule that it's making, the new DNA strand. Well, one thing about DNA polymerase is that it can't get this process started on its own. It requires the use of a starter molecule. And the starter molecule is this small RNA piece, which we refer to as a primer. It is RNA, which is why it's shown here as red, to indicate that it is different from DNA. And so that's our starting molecule in the red. And then DNA polymerase is going to keep adding on, shown here as a light purple, and it's going to keep going. What we see here is called the leading strand. The leading strand is going to be synthesized in one long continuous chain. Basically, we get that starter molecule, the primer, and then DNA polymerase just keeps going, and it sails along as soon as we get it started. So for the leading strand, synthesis, the actual making process or the direction of the movement of DNA polymerase, is going right into the fork, the replication fork. On the other strand, in this case the bottom strand that we're seeing, it doesn't happen like that. That strand is called the lagging strand. And on the lagging strand, the DNA is going to be synthesized in short segments that later on will have to actually connect and join together. The reason for this is that the DNA synthesis is actually moving in this overall direction. It's going away from the fork. And if you look here, we have a bunch of RNA primers. See all the red pieces that are being added? So we have to keep providing new starting places for DNA polymerase to come in and then extend. And then we put a new starting point and it comes in and it extends. Well, these little pieces that we are making, they're called Okazaki fragments. So you can see that term here on this slide. That's just the short piece of DNA. Eventually, we do want to join them together, but we also have to get rid of the RNA pieces. So you can see that the RNA piece is being cut out right here. DNA polymerase is able to cut it out or to remove it. It is a slightly different DNA polymerase, so if you look closely, you can see that DNA polymerase 3 was the one that did the main synthesis. DNA polymerase 1 is the one that's going to boot the RNA molecule off. Once we have the RNA off of there, then we make DNA to fill in the area where it was, and then we have another enzyme called DNA ligase, and DNA ligase is going to join the DNA pieces. Or in other words, it's going to join the Okazaki fragments together. So we've got DNA polymerase making the main, so we have primase, which is making the primer. We have DNA polymerase, which makes the DNA. And then we have DNA polymerase 1 here, which is going to remove the primer. So there's actually a lot going on at this particular replication fork when we're talking about the overall replication process. So this is looking at the lagging strand. The lagging strand, that one has the synthesis, as we said, that proceeds away from the fork. So again, the fork is there, and synthesis is actually going in this direction. So you've got the top strand that synthesis is going this way, and you've got the other one going in the other direction. We refer to that as anti-parallel synthesis. So DNA replication is bidirectional, and we could see that on that previous slide, that we do have those kind of DNA polymerases running in opposite directions. Well, another thing to understand is that we are talking about circular molecules, circular chromosomes in prokaryotes, and so when we pull the DNA strands apart from each other at the origin, you actually have two replication forks created there. And we will have synthesis going on both of the strands for this replication fork, and then we'll have it on both of the strands over here with this replication fork, so that ultimately it starts here and then it just goes and kind of continues around. And that's what you see happening right here. Just some terminology. The parent strands, these are the original strands. So that would be like the old DNA. And then the daughter strands, that's the new DNA. Now, as we start to separate and pull apart these DNA molecules, sometimes we get some over-twisting, some kinks that form in this molecule because it's a twisted molecule anyway. And so that excessive twisting, or supercoiling as we call it, that has to be relieved, taken care of by specific enzymes. And the enzymes that are going to be involved in that are gyrase, and then we also have topoisomerase, which is going to help deal with that particular problem. In the end, we start with one molecule that has one origin, and we end up with two daughter molecules, which are identical copies of each other, and they're also identical copies of the original molecule that we started with. Another thing that happens during this whole DNA replication process is that many times there will be some methylation that takes place. Methylation will occur to certain adenine and cytosine bases. And so what this means is that we have this extra chemical group. It's called a methyl group, and this is the methyl group if we were to actually look at it, and this is going to get added right there on top of the DNA. So why is methylation important? Why is this something that the cell is actually going to do during this DNA replication process? Well, one thing that the methylation is going to do is that it does act as a control of gene expression. And what we mean by gene expression is that this is one way for cells to regulate when they actually utilize different portions of the chromosomes. So the methylation is going to be involved in that. Typically, if something is methylated, so if that has been added on top of the DNA right there, then that will be something that gets used less. Another thing that methylation does is it plays a role in the actual initiation of DNA replication. It will help to protect against certain viral infections, and the way that it does that is that the viral DNA is not going to be methylated. So the cell can specifically notice that you have DNA there and it's foreign because it's not methylated. So it helps the cell to distinguish between normal bacterial DNA and then infecting viral RNA and infecting viral DNA. And then the last thing is that the methylation is going to help in the overall DNA repair process. We focused our time talking about prokaryotic DNA replication, but this is something that eukaryotes certainly have to do as well. In general, eukaryotic processes are going to be more complex than what we find in prokaryotes. So if we just go through and list some of the things that make eukaryotic DNA replication different from prokaryotic DNA replication, first on the list is that with eukaryotes, there's going to be more forms of DNA polymerase involved in the process. So there's just a longer list of ones that will be participating. There's also going to be lots and lots of starting points. With prokaryotes, we had a single origin for the bacterial chromosome. With the chromosomes in eukaryotes, you've got thousands of origins per chromosome. The reason for this is that there's just so much more DNA that has to be copied, and if we start in a bunch of different places, we'll be able to accomplish this process within the time frame that's allotted to it. Another thing is that the Okazaki fragments that are made during the process, they will still be made in eukaryotes, but these are going to be much smaller Okazaki fragments. And then also, if we talk about that methylation part, the methylation is only going to occur to the cytosine bases. So with prokaryotes, it happened to the A's and the C's. Here, it's only happening to the C's.